The topic of blood cell modeling is really at a point where there's a lot of sophistication in various models, and it's really at the time of trying to bring together these multiple scales from molecular to cellular to whole tissue. And certainly the role of mechanics and thermodynamics and fluid mechanics come into play into these types of problems in the cardiovascular system. I wanted to start off real quickly with an example. This is a pretty old example. Leonardo da Vinci was a really brilliant anatomist, but he was really a fluid mechanicist at heart. He really loved hydrodynamics. And what he appreciated was the geometry of the pulmonary valve, but he used the mathematics of his time, geometry, to make a hypothesis, to say what is the structure of the fluid flow. And he really had no way of understanding what is the flow in this, no way to visualize the flow at that time. And he predicted that there would be these recirculation eddies distal of the valve. And this is really a remarkable prediction to make 500 years ago, given the lack of any imaging. And in a patient with pulmonary hypertension, vortexes can actually be seen downstream of this valve. So this is really a remarkable synthesis, and I think this is what many of us try to achieve, is a synthesis between structure and function over various scales of physics. Now today, a computational fluid dynamics calculation can easily calculate trajectories, and we have very reliable constitutive models for how blood flows as a bulk liquid through complicated time-dependent geometries. And you can certainly see there's a mechanical bileaflet valve, a recirculation zone. So we can describe blood as a fluid at the macroscopic level, but really what our lab has been very interested in is describing blood as a reactive fluid, and to understand the reactions that occur inside of individual cells and how those cells can work cooperatively to address issues of bleeding and to stop bleeding. And sometimes those cells interact with each other during the course of disease, such as thrombosis in an artery. So in time, from a given patient, for example, we may understand the cells of that patient, we may understand the geometry of that patient, and we'd like to predict what is going on specifically at a location. Is this patient at risk of a heart attack? What would a heart attack look like in that patient with their reactive blood? And how might various drugs be effective for that specific patient? That's kind of the motivation of what we're trying to do. So blood clotting from a biochemical point of view, as it is assembled under flow conditions, various pieces of this have been very well studied over the last 50 years or so. Blood is easily obtained from the laboratory from humans. It's really the one tissue you can get from humans on a daily basis. So we know under flow conditions, if this is a damaged vessel wall, there's very rapid interactions, rapid bonding events, short-lived bonding events between GP1B and von Willebrand factor. Helps bring the platelet, the flowing platelet, and we'll probably see lots of talks in the next few days on platelets. Platelets will then interact with collagen, and this drives very strong signaling to activate integrins. So the cellular component here is very activatable, and this mediates firm arrest, but then we have this buildup of platelets, and this is autocatalytic. Diffusible species like ADP and thromboxane, thrombin are released by platelets, and you also have protease reactions triggered by the wall, the extrinsic pathway through tissue factor, and the intrinsic pathway or the contact pathway through factor 12A, ultimately polymerizing this mass together into fibrin. So this was an experiment we conducted a number of years ago where we perfused human whole blood over a microarray that had been printed with collagen spots, and we added increasing amounts of tissue factor, this is the trigger of the extrinsic pathway, to these spots. And what we saw over a very narrow dose response range, going from about 2 to 10 molecules of tissue factor per square micron, you go from basically a platelet deposition event to a full thrombotic response where you're polymerizing fibrin. So we wanted to model the accumulation of platelets at this location and also the production of fibrin through the assembly of the proteases in the coagulation pathway. So we've done modeling work, direct ODE modeling work on platelet signaling. I won't describe that work, but it is oftentimes a very tedious process to account for individual molecular events inside of a platelet as it responds to a stimuli. So in our earlier work, we wrote ODE models for how does a platelet respond to ADP through the P2Y1 receptor all the way to releasing calcium. And just that one pathway is about 200 parameters. You have about 130 kinetic constants and 70 initial conditions. There's a lot of work, one PhD student at a time, and we wanted to develop a quicker method because we know that a platelet in a clotting event responds to collagen, responds to thrombin, responds to ADP, responds to thromboxane. And the endothelium is producing things like prostacyclin to quiet the platelet down. So we developed a laboratory technique, we call it Pairwise Agonist scanning, where we use laboratory molecules that mimic the in vivo constituent. So this is convulsin, it's a venom that activates GP6 very strongly, the way collagen would activate it. We have used a stable analog of thromboxane to activate the thromboxane receptor. These are peptides to activate the thrombin receptors. ADP is easy to use in the laboratory. We have a stable analog of prostacyclin that we use to activate the IP pathway in platelets. So the platelet is functioning like a computer in that it's seeing a lot of signals in a time-dependent manner, and we want to know how is calcium regulated in this platelet, because calcium is very important in activating the integrins, causing granular release, activating enzymes that produce autocrinic factors, and changing the membrane of the platelet surface. So in the laboratory, this pairwise agonist scanning technique uses pairs of agonists, and we use each agonist at low, medium, and high concentration. And we stimulate the platelets with these pairs of agonists using a liquid handling reader, and we measure intracellular calcium going up and down. So this experiment gives us a data set. It only takes about an afternoon to do this. And all we have to do is isolate platelet-rich plasma and load it with dye. And we get these calcium traces of calcium going up and down. And usually people report these traces as time traces like this, but we have hundreds of them, so we show them as heat maps. And you'll see some agonists, if there's convulsin in there, the calcium stays elevated for long periods of time. Other agonists, like this prostacyclin mimetic, cause the platelet to quiet down, so you see a lot of blue here. A lot of the responses have been reduced. So there's actually 34,000 data points here. There's 154 calcium traces, and we use that to train a neural network model. So it's a very simple model. It's a data-driven model that tries to capture what is that individual's platelets doing in response to these agonists. And the neural network can be trained very accurately and recapitulate the data very precisely. And what we've shown I won't show the data here, but it's in the publication, is that we can actually predict how does the platelet respond to 3 agonists, 4 agonists, 5 agonists, 6 agonists. How does it respond to sequential stimulation? And what it's doing is it's building up a higher order prediction based upon the pairs, the pair data. So we can actually predict like synergies, trinary synergies, based upon the binary synergies. So what is a synergy? A synergy is a parameter that we can calculate from the data where we look at the response to pairs of agonists, agonists A and B used together, and we compare it to agonist A used alone or B used alone. So if it's simply additive, the synergy parameter would take on a value of 0. And if we're getting more calcium for the pair than the individuals, it would be a positive synergy. And if there's antagonism, that would be a negative synergy. So this is taking all those data points that I just showed and reducing it basically to a vector of numbers. And these are showing the synergy going from low, low dose to high, high dose for each pair that is tested. So we have a very complex phenotype that we're measuring, and the neural network, we can also calculate the synergy map here, and we do a very nice job of recapitulating this fairly large phenotype that we've measured. So our next step then is to incorporate this phenotype into a multiscale model. So we measured all these pairwise synergies in the laboratory, and this is just showing this set of numbers of synergies as a vector. We see that this vector is actually very representative of the individual. If we test people in replicate over a 2-week period, they self-cluster. We're seeing some interesting subclusters, how donors are different. But we want to then take this and embed that into a multiscale model. So let me also point out that we do a lot of microfluidic work, and our multiscale modeling is trying to simulate the exact conditions in this microfluidic device. So we take blood, and the work that I'll present now, there's no thrombin in this work. So we use a very strong inhibitor of thrombin, PPAC. So we're just looking at platelets deposit on a stripe of collagen, and the platelets are fluorescently labeled. They can release ADP and thromboxane, and they start building up with time. And you can see this buildup of platelets on the surface with time. If we add certain drugs, P2Y1 inhibitor will just pretty much reduce that deposition to a platelet collagen interaction without this secondary aggregation. There's tens of millions of patients on Plavix. That's a P2Y12 inhibitor, very potent inhibitor of secondary aggregation. So this is the type of data that we get, and in these microfluidic devices, we can dose response drugs, or we can run experiments in high replicate to get the statistics of the experiment. So what we did is we took blood platelets from various donors, and we did this pairwise agonist scanning. We don't have any thrombin in here. We're just looking at platelet function, and we stimulate the platelets with convulsin to activate the collagen signaling, a thromboxane mimetic, ADP, and in this experiment we're using iloprost. It's a stable analog of prostacyclin. And we measure intracellular calcium, and in the experiment we actually block this pathway, so we don't have any autocrinic signaling, and we're under very dilute conditions where ADP autocrine signaling is pretty minimal. So we are controlling the concentrations of these agonists in the experiment, all the pairs, and we're measuring calcium, and we train a neural network for each donor. And then this is the multiscale model. That neural network is embedded in each platelet, and each platelet is convecting and dispersing based upon a lattice Monte Carlo simulation for stochastic motions of a particle in the flow field. The particle itself has no effect on the flow field, but the platelet is really small in the moving flow field. But it's being jostled around, and that's captured by the dispersion coefficient, which is affected by the red blood cells. And there's a drift term driving the platelet toward the wall. So that's a drift velocity that's also in this Monte Carlo. And we've published on how to do convective diffusive Monte Carlo of flowing platelets, flowing particles. There's a boundary layer problem. These platelets release ADP and thromboxane, so we have to solve for the time-dependent release of ADP and thromboxane from these platelets. That's solving 2 PDEs by finite elements. This is actually a very costly part of the simulation. It's easily a third of the simulation, just following ADP and thromboxane. And then as this platelet mass grows, we solve the changing velocity field by lattice Monte Carlo. I'm sorry, by lattice Boltzmann. So the bonding model is fairly simple in this experiment. Our apologies to everybody doing single bond kinetics, but we have hundreds of platelets that are participating in the simulation, and we're accounting for bonding reactions that really involve hundreds to thousands of bonding pairs when a platelet sticks to another platelet. So we can keep track of the intracellular state. This is like an extent of reaction. This is just the integrated calcium response. So we can make a statement about how the calcium changes in each particle. We just integrate it with time. So that's the integrated calcium response. And then we map that onto a Hill function between 0 and 1. And then the on rate, this is a parameter that describes the deposition of platelets to collagen or platelets to other platelets. Depends on how activated the platelet is, which controls the receptor numbers. And then the off rate, if we're removing a platelet from collagen or removing a platelet from another platelet, depends inversely with the activation state. And also, we have a function of the shear rate. This is just a Bell exponential. Okay, so a very simple model of bonding, but it depends upon the activation state of the platelet. So this is the neural network training of donors 1, 2, and 3. And you can see we're training the neural networks very accurately. We're predicting the synergies very accurately with the neural network. And then this is a snapshot of the simulation for donor 1. And the red bar, I'll show you the movie in just a second, the red bar is the collagen. Platelets that are highly activated that's on a gray scale from resting black to highly activated white. They're highly activated. We have a boundary layer set up for acute release of ADP from granule dumping, and then also the synthesis of thromboxane in this boundary layer. And then as the surface becomes rough, the shear rate on the surface can change dramatically by about a factor of 5. So we may start the experiment at a venous level of shear rate, but at the peaks here, we're actually approaching arterial levels of shear rate. So this is what the simulation, an individual simulation looks like. It takes about an hour to run this on a PC. It's a very fast simulation. And we can run hundreds a night at the University of Texas on their supercomputer. This is for donor 1. The platelets are activating. The surface is really becoming much more reactive now. We're developing a boundary layer of ADP. ADP is released very acutely, so you'll see these flashes of pre-synthesized ADP. Thromboxane is synthesized more slowly, so its boundary layer develops more slowly but is a little more uniform. And then there's a characteristic roughness that develops in these simulations. These aggregates grow preferentially. Once an aggregate starts growing, it's going to grow more rapidly than another zone that is not as accessible fluid mechanically. And the characteristic aggregate size here is about 40, 50 microns in size. So we run the exact same experiment with the same blood from the same donor, and this just shows this is sped up a little bit, but the fluorescent platelets depositing on this collagen stripe, no thrombin present, and you can see this is why tens of millions of people take aspirin, reduces platelet deposition, endomethacin, and also a P2Y1 inhibitor. So we can modulate the pharmacology in this experiment, all in the same experiment. So we're comparing now the simulated platelet deposition for a donor based upon their platelet measurements, the neural network that we trained. And we see that donor 1 always makes larger clots in the experiment than donors 2 and 3. And this is like n equals 16 clots. We have high replicate with these microfluidic devices. The computer simulation, the only thing different in these simulations is the platelet neural network. All the physics is the same. We see in the computer simulation that donor 1 makes larger clots than donors 2 and 3. And the simulation has no trouble predicting the rank-ordered potency of these various drugs, aspirin, endomethacin to take out thromboxane, MRS-2179 to take out ADP signaling, iloprost. We should all have healthy endothelium bathing our platelets in prostacyclin, very potent inhibitor. So you can see in the presence of iloprost, all these donors are the same. The neural network has no trouble making that prediction. So this is in an experiment where we start the flow rate at a venous level of flow. Same neural network model. We run experiments at higher levels of, these are arterial levels of shear rate, and the neural network can do a very nice job of predicting not just the extent of platelet deposition at venous and arterial levels, but also the error bars. You know, the statistics of this experiment in the Monte Carlo are also accurately predicted. So what was interesting, when we ran this experiment, we noticed that donor 3 was not responsive to aspirin. And this is a very controversial topic in the cardiology literature. There's 50 million people in the United States taking an aspirin every other day. That's a lot of people walking around taking aspirin. And it's not known who derives benefit from that pill of aspirin, who is at risk for GI bleeding, or who has no benefit at all. And here, and the cardiologists often say, everybody responds to aspirin. Some patients don't take their aspirin. Okay. Well, we know aspirin is present in this experiment because we added it in the test tube. It is absolutely present, as is endomethacin. This patient is not responsive. We went back to the data and saw that they weren't responsive to this thromboxane mimetic. That led us to hypothesize that there was a mutation in the thromboxane receptor. We sequenced the mutation, sure enough, found a point mutation that when you express, you lose loss of function. So that's a heterozygote mutation, pretty rare. This is the only donor out of about 25 that's not responsive to U46619. But it is a new mechanism for aspirin resistance. It's as if this patient is always on aspirin. These types of mutations have been described before, but this is the first report of this particular point mutation. It lends itself to a very mild bleeding genotype. So we're interested in aspirin resistance. I won't talk much about this, but we are using these microfluidic devices to actually characterize sensitivity to aspirin. And we've actually found 7 subjects out of 28. We know aspirin is acetylating their cyclooxygenase, but in this functional assay, we don't see any inhibition of secondary aggregation. Okay. So let me finish up talking about thrombin. Everything that I've just shown is about platelet physiology, but we know that as platelets deposit on the wall, there's thrombin production. And thrombin is a very potent platelet activator, but also polymerizes fibrin. So this is a fluidic device where we have blood flowing over collagen. This collagen contains tissue factor, and we can independently control the wall shear rate and the pressure gradient driving permeation, Darcy flow permeation. And this is a typical clot structure that we develop. These are the platelets, and the fibrin is formed in this very thin layer where the tissue factor is at the interface between the blood and the tissue factor. And we're pulsing fluorescent solutes through this media to get the permeability of this structure. And in the experiment, if we stop the flow to switch to the fluorescent dye, which we don't do anymore, but something very striking happened when you stop the flow. This clot structure contracted like a muscle very rapidly. So there's a very low level of contraction occurring under flow conditions as the clot is formed, but you stop the flow, and all of a sudden you see this massive contraction, and we can map that contraction. And what is occurring is that there's a quorum sensing going on. The platelets are releasing ADP and thromboxane. I showed you those boundary layers in the simulations. And this ADP and thromboxane is being washed away. But if you stop the flow, all of a sudden that ADP and thromboxane builds up very rapidly and drives this contraction. If you use blebbistatin, you'll block the contraction. If you use receptor antagonists, you'll actually prevent. We've also detected a calcium mobilization. You'll also prevent that calcium mobilization as well as the contraction. So a blood clot is a flow sensor. There may be some mechanobiology in there, but primarily it's all about quorum sensing. It's all about this coupling between transport and platelet retraction. So in discussing thrombin, we have very, very good models of how thrombin is formed isotropically in a test tube. It's much harder to incorporate those kinetics into a flow model because solving ODEs is incredibly rapid and solving PDEs is incredibly computationally intensive. But we do have good kinetic descriptions of how thrombin is formed on platelets in the presence of tissue factor or contact activation. The FDA is actually using this model right now in calibrating some of their work on blood products. But we can incorporate tissue factor in our experiments and drive the production of thrombin and drive the production of fibrin. Normally, you can't detect this unless you add tissue factor to drive the thrombin production. And what's important in these types of experiments, when you have thrombin made, the clot can actually grow sufficiently to block the channel. Now, if you're operating in a constant pressure drop mode, that occlusion will stop flow, just like blood flow stops in a blocked coronary artery. However, most experimentalists will just set flow rate on the pump as constant flow rate. And what happens there is the shear rate will skyrocket into tens of thousands of reciprocal seconds and ultimately break off this clot. So it's a measurement of clot strength, really, if you operate under constant flow. So if you have thrombin driving the growth process of the structure, it's important to operate in this pressure relief mode. But we've done a whole study of clot breakup and clot growth in the presence of fibrin production. And what we've seen is that clots without fibrin can withstand a couple thousand reciprocal seconds. Once you have fibrin there, they can start to withstand tens of thousands of reciprocal seconds of wall shear rate. So the fibrin imparts tremendous stability. Now in these experiments, as the clot is growing across the channel, the shear rate skyrockets. And there's actually a change in von Willebrand factor biology is that VWF, this 20 million molecular weight entity, can, as a polymer, can uncoil in the presence of these extreme flows, and we've characterized this. So you can see that in a clotting event, you have thrombin-driven processes, you have platelet-driven processes with retraction, and if the clot grows extreme enough in size, you'll get into this von Willebrand factor-mediated regime where you have a change in the molecular conformations of von Willebrand factor. So thrombin is a little difficult to measure because it's made, it's very potent, but it floats away. And we developed a sensor that's linked to a platelet that can detect this thrombin activity. And this has really helped us localize where the thrombin is. This is in a mouse model, laser injury that Skip Brass did. The thrombin we see is very near the injury site, not in these outer regions of the shell. It tends to stay in the core, which is very stable, but these outer regions are much looser structure, and these outer regions is where the ADP and thromboxane play much more important signaling role. If you have permeation in, say, for example, a hemostatic event, permeation across that structure into the interstitium, it will control where the thrombin is actually located. So we have pretty good correspondence between what you see in a mouse model in terms of these multiple zones of chemical activation, thrombin activation, and what we see on our in vitro models with human blood. So it's provided a nice opportunity to go back and forth. So where we are now is we're trying to really incorporate very detailed patient-specific descriptions of platelet signaling with very detailed descriptions of the kinetics of thrombin production, ultimately to include platelet retraction mechanisms and also fibrin stability. Let me just acknowledge some of the students who have been involved. Keith Neves got us started in microfluidics over the years. Manash Chatterjee and Jeremy Purvis developed the Pairwise Agonist scanning techniques. And Matt Flam did a lot of the work with the lattice kinetic Monte Carlo for the stochastic particle transport. So I thank you for your attention.